So yesterday we talked about these kind of spectra, and every spectra to the top left is the carbon 13 NMR, and there is a proton to the right, proton spectra for this compound. This is from my database, discovered molecules, and this is one of them. The lower structure is the X-ray, and we can see exactly how many carbons, how many rings, or what type of functional groups, how many central metals, everything from the X-ray. So the X-ray being obtained from the pure sample, as we call it, crystals. So when there is a crystals, and these crystals are like need to be examined from a specialist, and we build up our experience by time. Thanks for colleagues before us. They showed us how to understand these things in chemistry, which we couldn't like find them in books before. So this is once we are within a scientific group, everyone has experience, and thanks for the professors and the supervisors, which they are experts, and they give us the overall plans and advices and scientific tips and information. But the details, the practical, you can find it from the colleague of yours, the ones before you. So it's like transferring knowledge from person from within the group, from person to person. This is very easy to get it ready before you go to books and investigate and see, and always practical experience. You cannot easy find it from books. You need to find it from the work itself, from people in the working area, and then you can learn from them straight away with short time, within a short time. So we were like PhD research, very good. I remember like I was far away from chemistry for 10 years, and then I returned. I had my bachelor degree from my own country, and then I moved after years, after 10 years, I moved to Ireland, and in Ireland, uh, I was good in English. That was very good approach, or it was easy to get inside the university. And then from there, I needed to learn chemistry language. So there is a difference in normal language and the chemistry, which we use like chemistry terms and words. And so where can you find these? You can find them ready in the colleges, in the universities, and these are very like experienced people working in the industry for like generation after staff after staff. So they gained experience. Every one of the teachers, of the lecturers, they were there for 20 years, 30 years. So they had great informations to pass them to us. And we were students. We came know little things, and they gave us everything within short time. So as I remember these structures, this is the X-ray. Above is the carbon 13. To the right is the proton NMR. How can we understand these peaks? Every peak related to functional group. What are these noise? The noise, because it's the carbon, so we can see such a noise, but the clean spectra, we can see it from the proton, because the number of protons they are, we call it unshielded. They are outside from each molecule. So if you see to the down, I can show you like here. Uh, so these are two cobalt centers. There is a bridge of oxygen. There's a nitrogen, three types of bonding nitrogens. And from outside, there is a ring, penta ring, and carbon. And here, these small, tiny circles are hydrogens. So here, like this group, this ring has how many hydrogen? 1, 2, 3, 4, 5, 6. Here, the benzene ring, which how many hydrogen or free? Is it a benzene? No, it's a imidazole ring. So the imidazole rings, they have 2 nitrogens and 4 hydrogens. So it's easy to identify, not that easy, but I'm saying for, to identify the hydrogen is easy. So we have only 4, and these are from outside. They can get excited, absorb energy, and we can find the peak for each hydrogen. So this, like, how can we find the peak? How can we know what is this small peak related to what? This is the higher one, and so on the range of the NMR, there is a value for this peak. We bring incident line from the top of the peak till here, and you can decide what's the ppm, part per million. And next one will be different. So this is a broad peak. This is a sharp peak, small broad. Each one of, like, most of the broad peaks related to the water hydrogens. So I remember that the broad one is related to water. And so we can know there is a water here or somewhere. So, like, understanding the structure and each hydrogens related to which functional groups. So here we have a carbonyl, dicarbonyl, or carbonyl dibridged, or bidentate. So with the carbon here, so even you have the X-ray, but you need to provide IR, NMR, UV vis, if it was colored. So it's magnetic properties. If all the data should be collected for each molecule and then publish it in a paper. And once it's related to the manufacture of this product, can be useful for antibacteria, antivirus, anti fungus or something. So they are very good mimicking or behaving similarly with the enzyme active sites, so related to bacteria or produced by bacteria. So here, as I said, we like finished the PhD within 3 years, and we discovered my research was discovered more than 100 new compounds, and they are, if being checked more in biology, they will show the behavior of preventing kind of bacteria, which type of bacteria to stop producing enzyme, like hurting the stomach and the digestive system. So now I go to coordination complexes in living systems. So the most popular ones is porphyrins. Porphyrin rings, they are in very famous compounds or super molecules. When I say super molecules, means high atomic weights of such. So they are not small molecules of 2, 3, or 10 or 20 atoms bonded together. No, these are millions in the one unit cell. So in the living systems, in biology, we can find porphyrins, hemoglobin, chlorophyll, vitamin B12, and so on. So porphyrins and hemes, this is the ring of the porphyrin ring, consists of 4 nitrogens. They are ready for bonding. And the rest you will see here, penta, 4 penta circles or cycles, and you can have C20, H14, and 4. That's the porphyrin ring. So there is a bridging, and there is rings as well. So now, if you put the iron in the middle, they will become heme. So this is the heme with the porphyrin ring, and the heme came in the middle and been bonded to 4 nitrogens of the porphyrin. So now these planar molecules have a hole in the center in which a metal can coordinate or reside. Porphyrin ring and iron structure, similar to this. So this is it. We draw it differently or similarly. So 4 nitrogens, 2 with hydrogen, other 2 bonded to 3 bonds. As you see, every nitrogen is 3 bonded. And here are the penta rings. And here are, maybe we call it a bridging aliphatic ring. And then when the iron comes inside in the center of it, so they will be bonded to the iron. Why, if we come here, why do we need to have D metals? Some critical enzymes in our cells are metalloproteins, giant biomolecules which contain metal atom. These metalloproteins control key life processes such as respiration and protect cells against disease. Hemoglobin is a metalloprotein which contains an iron atom and transport oxygens throughout living systems. Vitamin B12 prevents pernicious anemia, contains a cobalt atom which gives the vitamin red color. Now, reversible addition of oxygen to hemoglobin. You can see here the lungs. The lungs absorb oxygen, and we can see that there is a mechanism for this. So here I can show you how this mechanisms, how oxygen or dioxygen, bioxygen bonded to the iron in the center, and this bonds of the iron to the left structure. Once the oxygen comes from the breathing, so the iron is ready to bond to this bioxygens or oxygen gas. What will happen? The mechanism by which oxygen is carried throughout the body is provided in papers. I can explain to you. So this is the bond. Once the oxygen is bonded to the iron, then it will be transferred to the, like this is in the lungs. So the hemoglobin with the oxygen will go to the cells, and from the cells it will release the oxygen and bond to the, so the porphyrin itself flat, but once bonded to the oxygen will change the flatness, and when releasing the or release the oxygen and uh, link to carbon dioxide, the ring or the porphyrin ring of the hemoglobin will be changed. And this is very unique. How is this happening? Why? Because of the bond, new bond to the metal, to the iron. That's make the behavior of the whole structure of the porphyrin of the ring be such alike. So it's like flexible and or waveable. So then once the releasing oxygen inside the cells, then it will uh, bond to the carbon dioxide and then bring it back to the lungs, then release it, release the carbon dioxide, and then receive another molecules of oxygen and so on. So oxygen released in the body cells and carbon dioxide carried out. Vitamin B12, cobalamin, very important. And the cobalamin this is the structure to the right is very big. And there is a cobalt metal center to the left. You can see same structure. And the cobalt, there are similar porphyrin ring for nitrogen bonded to the cobalt. But from the top and bottom, there is a different connection to a different functional groups. And vitamin B12, most important functions involved in production of red blood cells. Look how important is cobalamin. Production of red blood cells, normal functioning of brain and nervous system, synthesis and regulation of DNA and fatty acids. Only where can we find cobalamin? How can we produce it? Only bacteria, archaea, have the enzymes required for synthesis of such vitamin. So vitamin B12 is not produced by our body or any plant or anywhere. Only bacteria can produce the enzyme which required synthesis of vitamin B12. So it's a cobalt. You see the formula here underneath. Cobalt and 62 carbon, 88 hydrogen, 13 nitrogen, 14 oxygen, 1 phosphor. And then outside you have a counter ion, is CN, cyanide. So this is usually taken from tablets. We can purchase from or buy from the pharmacist or shopping centers. Companies are making these vitamin B12. They are very important to be taken every day. Chlorophyll, very important porphyrin type molecule found in plants, converts solar photons into food energy. So what's the metal center of inside the chlorophyll is magnesium. Same thing, we have porphyrin ring around the magnesium for nitrogen bonded. To the right side, I'm looking. And to the left side, it's a bulk or the X-ray. To the left, we can see the magnesium in green, the 4 nitrogen in blue, and then surrounded by the porphyrin ring. And this porphyrin ring contains different functional groups, different from the other species we talked about just in a second. So every porphyrin has like functional groups around it. This is like you see outside there is types of other groups linked to the porphyrin. This is porphyrin naked to the left. This is to the right. You can see COOH at the bottom, two of them. And at the top, there is some CH2, CH3 groups. So they are different in hemoglobin. But in the other, here you can see the porphyrin naked, but there is a protein around it. Look at how big is this porphyrin joint functional groups. So as we said, talk about vitamin B12, very important. Chlorophyll is very important for plants, for animals, or yeah. So they do photosynthesis, again, absorb carbon dioxide, release oxygen, very important in the green system. And animals, human being, other species rely on chlorophyll for the life cycle. So this is the end of this section, and we can stop this and go to the next